DNA, or deoxyribonucleic acid, is a molecule that contains the genetic instructions for the development, functioning, growth, and reproduction of all living organisms. It is often referred to as the building blocks of life. DNA is a double-stranded helix structure made up of nucleotides, which consist of a sugar, deoxyribose, a phosphate group, and one of four nitrogenous bases, adenine A, thymine T, cytosine C, and guanine G. These bases pair up in a specific manner, A with T and C with G, forming complementary base pairs. This complementary base pairing is crucial for the accurate replication of DNA during cell division. Interestingly, the amount of adenine is always equal to the amount of thymine, and the amount of cytosine is always equal to the amount of guanine in a DNA molecule, a concept known as Chargaff's rules. DNA is organized into structures called chromosomes, which are located in the nucleus of eukaryotic cells. In prokaryotic cells, DNA is found in the nucleoid region. The human genome, which is the complete set of DNA in a human, contains approximately 3 billion base pairs. DNA carries the genetic information in the form of genes, which are specific sequences of nucleotides that code for proteins. Understanding the structure and function of DNA is fundamental to unraveling the mysteries of genetics and heredity. The genetic code is the set of rules by which information encoded within genetic material, DNA or RNA sequences, is translated into proteins by living cells. This translation is carried out by codons, which are sequences of 3 nucleotides that correspond to a specific amino acid or a stop signal. There are 64 possible codons, with 61 codons encoding for amino acids and 3 codons serving as stop signals to terminate protein synthesis. The genetic code is degenerate, meaning that most amino acids are encoded by more than one codon. For example, the amino acid leucine is encoded by 6 different codons. However, the start codon, AUG, serves a dual purpose as both a start signal for protein synthesis and as the codon for the amino acid methionine. This versatility is crucial for the accurate initiation of protein translation. Interestingly, the genetic code is nearly universal across all living organisms, from bacteria to humans, with only a few exceptions. This universality suggests a common evolutionary origin of life on Earth. The discovery of the genetic code was a monumental achievement in molecular biology, revolutionizing our understanding of how genetic information is stored and utilized by cells. The deciphering of the genetic code paved the way for advancements in genetic engineering, biotechnology, and personalized medicine. Scientists continue to study the genetic code to uncover its intricacies and potential applications in various fields. The genetic code and codons play a central role in the process of protein synthesis, which is essential for the structure, function, and regulation of all living organisms. Understanding the genetic code provides insights into the complexity and diversity of life, highlighting the remarkable precision and efficiency of cellular processes. As we delve deeper into the secrets of the genetic code, we unravel the mysteries of life itself, unlocking new possibilities for scientific discovery and innovation. Transcription is a fundamental process in molecular biology that converts the genetic information stored in DNA into RNA molecules. This crucial step is the first stage in gene expression, where the genetic code is transcribed from the DNA template to produce messenger RNA, mRNA. The process of transcription is carried out by a molecular machine called RNA polymerase, which recognizes specific regions on the DNA known as promoters. Promoters serve as signals to initiate transcription and determine which genes are transcribed and at what rate. Interestingly, only one strand of the DNA double helix, known as the template strand, is transcribed during this process. The other strand, called the non-template or coding strand, serves as a reference for the sequence of the resulting RNA molecule. RNA polymerase moves along the DNA template, unwinding the double helix and synthesizing a complementary RNA strand by adding nucleotides according to the base pairing rules. Adenine in DNA pairs with uracil in RNA, cytosine pairs with guanine, and so on. As RNA polymerase progresses, it generates a single-stranded RNA molecule that mirrors the sequence of the non-template DNA strand. Transcription is a highly regulated process, with various factors influencing when and how genes are transcribed. Transcription factors, which are proteins that bind to specific DNA sequences, play a crucial role in controlling gene expression by promoting or inhibiting transcription. Additionally, epigenetic modifications such as DNA methylation and histone acetylation can impact the accessibility of DNA to the transcription machinery, thereby influencing gene transcription. The completion of transcription results in the formation of a primary RNA transcript, which undergoes further processing to produce mature mRNA. This processing involves the removal of non-coding regions called introns and the joining of coding regions called exons through a process known as RNA splicing. The mature mRNA molecule is then ready to be translated into a protein during the subsequent stage of gene expression, known as translation. Translation is the process by which the genetic information encoded in mRNA is decoded to synthesize proteins. This essential step in gene expression occurs at cellular structures called ribosomes, which serve as the molecular machinery for protein synthesis. The process of translation involves three key players: mRNA, transfer RNA, tRNA, and ribosomes. mRNA carries the genetic code from the DNA in the nucleus to the cytoplasm, where protein synthesis takes place. tRNA molecules act as adapters that bring specific amino acids to the ribosome based on the codons present on the mRNA. Each tRNA molecule has an anti-codon region that pairs with the complementary codon on the mRNA, ensuring the accurate incorporation of amino acids into the growing protein chain. The ribosome serves as the site where mRNA and tRNA interact to facilitate the assembly of amino acids into a polypeptide chain. The ribosome has two subunits: the small subunit that binds to the mRNA and the large subunit that catalyzes the formation of peptide bonds between amino acids. The process of translation can be divided into three main stages: initiation, elongation, and termination. During initiation, the ribosome assembles on the mRNA near the start codon, AU, with the help of initiation factors. The first tRNA carrying the amino acid methionine binds to the start codon, marking the beginning of protein synthesis. In the elongation stage, the ribosome moves along the mRNA, reading each codon and recruiting the corresponding tRNA molecules with the appropriate amino acids. Peptide bonds form between adjacent amino acids, leading to the elongation of the polypeptide chain. The process continues until a stop codon is reached, signaling the termination of translation. Interestingly, the genetic code is redundant, meaning that multiple codons can code for the same amino acid. This redundancy provides a level of robustness to the translation process, as errors in the mRNA sequence or tRNA binding can be tolerated without affecting protein function. Additionally, the speed of translation can vary depending on the sequence of the mRNA and the availability of resources within the cell. Certain sequences, known as Shine-Dalgarno sequences in prokaryotes and Kozak sequences in eukaryotes, help to facilitate the efficient initiation of translation by promoting ribosome binding to the mRNA. The accuracy of translation is crucial for the proper functioning of proteins, as errors in protein synthesis can lead to misfolded or non-functional proteins. Quality control mechanisms, such as the proofreading activity of the ribosome and the action of molecular chaperones, help to ensure the fidelity of translation. The completion of translation results in the release of the newly synthesized protein from the ribosome, ready to carry out its specific biological function. The intricate process of translation highlights the remarkable precision and complexity of cellular machinery, showcasing the elegant interplay between genetic information and protein synthesis. Ribosomes play a crucial role in protein synthesis, serving as the cellular machinery responsible for translating the genetic code into functional proteins. These complex structures consist of two subunits: a small subunit that reads the mRNA sequence, and a large subunit that catalyzes the formation of peptide bonds between amino acids. Ribosomes are composed of ribosomal RNA, rRNA, and proteins, with the rRNA molecules providing the catalytic activity necessary for protein synthesis. Interestingly, ribosomes are found in all living cells, from bacteria to humans, highlighting their evolutionary conservation and importance in cellular function. The process of translation begins with the binding of the ribosome to the mRNA near the start codon, followed by the recruitment of tRNA molecules carrying specific amino acids. As the ribosome moves along the mRNA, it facilitates the assembly of amino acids into a polypeptide chain, guided by the complementary base pairing between codons on the mRNA and anticodons on tRNA. Ribosomes can synthesize proteins at a remarkable speed, with some ribosomes in prokaryotic cells producing up to 20 amino acids per second. Additionally, ribosomes can coordinate the synthesis of multiple proteins simultaneously, forming complexes known as polysomes. The fidelity of protein synthesis is maintained by the proofreading activity of the ribosome, which helps to ensure the accuracy of codon-anticodon interactions. Ribosomes are dynamic structures that can adapt to changing cellular conditions, responding to signals that regulate protein synthesis. Overall, ribosomes are essential molecular machines that play a central role in gene expression and the production of proteins critical for cell function and survival. Genetic mutations are alterations in the DNA sequence that can have a wide range of effects on an organism. These changes can occur spontaneously during DNA replication, as a result of environmental factors such as radiation or chemicals, or due to errors in DNA repair mechanisms. Mutations can impact gene structure, function, and regulation, leading to diverse consequences. One common type of mutation is a point mutation, where a single nucleotide is substituted for another. This can result in the production of a different amino acid in the protein sequence, potentially altering its structure and function. For example, a point mutation in the hemoglobin gene can lead to sickle cell anemia, a genetic disorder characterized by misshapen red blood cells. Another type of mutation is a frameshift mutation, where nucleotides are inserted or deleted from the DNA sequence, causing a shift in the reading frame during translation. This can lead to a completely different protein being synthesized, often with non-functional or deleterious effects. Interestingly, some mutations can be beneficial, providing an evolutionary advantage to an organism. For instance, mutations in the gene encoding lactase, the enzyme that breaks down lactose, have arisen independently in human populations that practice dairy farming, allowing them to digest milk into adulthood. On the other hand, mutations in tumor suppressor genes, or oncogenes, can contribute to the development of cancer. These mutations can disrupt normal cell growth and division, leading to uncontrolled proliferation and tumor formation. Understanding the effects of genetic mutations is essential for diagnosing and treating genetic disorders, as well as for developing targeted therapies for diseases such as cancer. Advances in genomics and gene editing technologies have enabled scientists to study mutations at the molecular level and explore potential interventions. By unraveling the complexities of genetic mutations, researchers aim to improve human health and well-being through personalized medicine and precision genetic therapies. Regulation of gene expression is a complex process that allows cells to control when and to what extent genes are turned on or off. This regulation is essential for maintaining cellular functions, responding to environmental cues, and ensuring proper development and differentiation. One mechanism of gene regulation is through transcription factors, proteins that bind to specific DNA sequences and either promote or inhibit gene transcription. These transcription factors can act as activators, enhancing gene expression, or as repressors, suppressing gene activity. Another level of gene regulation involves epigenetic modifications, which can alter the accessibility of DNA to the transcription machinery without changing the underlying genetic sequence. DNA methylation and histone modifications are examples of epigenetic changes that can influence gene expression patterns. Interestingly, gene expression can also be regulated post-transcriptionally through processes such as mRNA splicing, stability, and translation efficiency. MicroRNAs, small RNA molecules that can bind to mRNA and inhibit protein synthesis, play a key role in post-transcriptional gene regulation. Cells employ intricate regulatory networks to coordinate gene expression in response to internal and external signals. For example, during development, specific genes are turned on or off in a precise temporal and spatial manner to ensure proper tissue formation. Dysregulation of gene expression can lead to various diseases, including cancer and metabolic disorders. Studying the mechanisms of gene regulation provides insights into normal cellular processes and disease pathology, offering potential targets for therapeutic interventions. Biotechnology and medicine have benefited greatly from advancements in genetic research. In biotechnology, Genetic engineering techniques are used to produce valuable proteins, such as insulin and growth hormones, through recombinant DNA technology. Gene editing tools like CRISPR-Cas9 offer precise ways to modify genetic sequences, holding promise for treating genetic disorders. In medicine, personalized genomics allows for tailored treatments based on an individual's genetic makeup, improving outcomes and reducing side effects. Pharmacogenomics utilizes genetic information to optimize drug therapies, ensuring efficacy and safety. Additionally, genetic testing can identify predispositions to diseases, enabling early intervention and preventive measures. The intersection of genetics, biotechnology, and medicine continues to drive innovation and revolutionize healthcare, offering new possibilities for diagnosis, treatment, and disease prevention.